Okay, so today we are here for the defense of Jerry, who's been in the lab since 2009. Before Jerry joined our lab, he actually he did his master's also in bioinformatics in the City University in Hong Kong, and he published a bunch of papers. He's worked on many projects in the lab. He was like one of probably the main person doing all of our methods for sequence analysis for many years, and since that time he's gotten into gene-gene interactions and other problems. Unfortunately for us, for science also, Jerry decided to go to Google after his PhD, so he'll be working there starting tomorrow, I think is his start date. It's been great having you in the lab, and I'm looking forward to hearing about some of your work.
Thanks for the introduction from Eleazar. So today I'm going to talk about the research projects conducted during my PhD study. And the title of my talk today is Efficient Statistical Models for Detecting and Analyzing Human Genetic Variants. So first, please allow me to briefly introduce some basic knowledge in human genetics. The DNA sequence is the genetic information carrier of human and other species. The DNA sequence of a human is composed of a series of 3 billion nucleotides, including A, C, T, and G. Here we show an example of the DNA sequence of a human. The human shares 99.9% of the DNA sequence, and there's only 0.1% difference between individuals. We call these differences genetic variants or genetic variations. The genetic variants can range from single nucleotide difference to large segments in the genome. And the difference on the single nucleotide level is called Single Nucleotide Polymorphisms, or SNP. So here we have 2 sequences. In the second sequence, it only differs from the first sequence by 1 nucleotide. So this is an example of the SNP. The genetic variance on the segment level is called structural variation, including insertion, deletion, copy number variation. In my study, I mainly focus on the copy number variation, and I will give some more detail about the copy number variation later in this talk. The genetic variants play an important role in regulating human disease and traits such as height, body mass index, and so on. So now we have 2 research questions. The first question is how to discover the genetic variants, including SNPs and structural variations. And the second question is which of the genetic variants are related to a particular disease or the phenotype of interest. Based on these 2 questions, my PhD work can be categorized into 2 groups. The first group is to detect the genetic variants, and the second group is to analyze the genetic variants. Today, I will mainly talk about 3 projects. The first one is copy number variation detection from high-throughput sequencing data. And the second project is gene-by-gene interaction detection. And the third project is IBD association mapping for quantitative traits. Now, I'm going to talk about the first project, which is the CNV detection from high-throughput sequencing data. The copy number of a DNA region is the number of copies of the region in the DNA sequence. And the CNV are the regions where the copy number varies between individuals. In this sequence, the region B, which is shown in blank, appears once in this sequence. So the copy number of region B is 1 in the first sequence. And in the second sequence, the region B, we can see it appears twice. So the copy number of region B in the second sequence is 2. There's a difference between the 2 sequences, so this is a CNV region. Many methods have been proposed to detect CNV using the high-throughput sequencing data, and the traditional methods will follow the following framework. So we have the genome of the interest, which is the donor genome. However, due to the limitation of technology, we are not able to directly observe the donor genome. And we can use the high-throughput sequencing technology to generate short segments from the donor genome. The short segments are called reads, and use the sequencer, we can generate reads uniformly from the donor genome. And then, in the next step, we can map the reads to a reference genome, which is already known. So the reference genome is shared by the human beings. And we use an algorithm called mapper to map the reads to the reference genome. Then we can compute how many reads are mapped to each position in the reference genome. The number of reads on each position is called the read depth. In this example, we can see the black region appears twice in the donor genome, so the reads from these two regions can all be mapped to this black region in the reference genome because the sequence are very similar. And then, because we assume that the reads are uniformly generated, so the number of reads mapped to this region should be expected to be twice of the number of reads mapped to the other regions. So this indicates the presence of a CNV. However, there's some challenge for this traditional method. The challenge comes from the read mapping uncertainty. For many reads, they can be mapped to multiple positions in the reference genome. These reads are called the multiple mapping reads. For these multiple mapping reads, the mapper cannot determine where to align the read in the reference genome because for these two positions, they have exactly the same sequence. So we don't know where to put it. And the traditional method will either discard all those multiple mapping reads or just randomly choose one position for those reads. Of course, this won't be correct. In my work, I designed the EM algorithm to handle this case. So now I will use a toy example to show why the traditional method fails and how my method works. So in this example, we have the reference genome. In the reference genome, we have two regions, A and B. They are very similar, and they only differ by one single nucleotide. In the donor genome, the region B is duplicated. So the copy number of region B in the donor genome should be 2, and the copy number of A should be 1. Again, we generate reads from the donor genome using the high-throughput sequencing technology. And here I only show the reads from these three regions. Then we map the reads to the reference genome. And now we see that for read R1, it can be mapped to region A and region B with equal quality. And we don't know where to really align the reads. So I show this read in red color. And for read R3, because it contains a C, and it will only be mapped to region A, not region B. So I show this read in black. In this example, we have 6 multiple mapping reads and 3 unique mapping reads. So if we discard all those multiple mapping reads using the traditional method, the read depth of this region A and region B will be lower than the other regions, and the method will predict them to be deletion. Of course, this is not correct. And let's see what will happen if we randomly choose one position for those multiple mapping reads. We will see that the reads will be assigned to region A and region B equally. So the read depth of each region will be 1.5 times of the other regions. However, the true solution should be that the read depth of region B is twice, and the read depth of region A is the same. So this method is also not correct. So I proposed an EM algorithm, which is called the CNVeM, to handle this challenging case. In my method, I first get an initial guess of the copy number for each position in the reference genome. And then for those multiple mapping reads, I assign the read to one of the mapping positions according to the current guess of the copy number. So I will show by example how the method works. Initially, we guess the copy number for each position to be 1. So in the first iteration, for the multiple mapping reads, we assign it to region A and region B with equal probability. And we have 3 multiple mapping rates in region A and 3 multiple mapping rates in region B. And the read depth of region A is 4 and the read depth of region B is 5 because there are some uniquely mapping reads. So we can update the copy number of region A to be 1.3 and the copy number for region B to be 1.7. This is the new copy number as estimation. Sorry. And then in the next iteration, we reassign the multiple mapping reads according to the updated copy number. This process is going on iteratively until the method converges. So in the next iteration, we see only 2 red reads is mapped to region A and 4 reads is mapped to region B. So now we updated the copy number of region A to be 1, the copy number of region B to be 2. This is the true solution and the method converges. So we handled this challenging case. We saw that our method has a high accuracy by experiment. We apply our method to a real data, which is the sequencing data of chromosome 17 of mouse strain AG, published by the Sanger Institute. In their paper, they report 31 copy gain regions, 43 copy loss regions, and 416 deletion regions. In their method, they didn't predict the exact copy number. However, our method is able to tell what is the copy number, whether it's 1, 2, 3, or 4. And our method reported 44 copy gain regions, and there are 28 overlap with their method. And for the copy loss regions, because we can tell what is the copy number, so copy loss and deletion is actually in the same class for our method. We reported 355 copy loss regions, and they all overlap with at least one of the copy loss regions or deletion regions. And we missed 29 of them. I checked each of these 29 regions and found that they are all really small regions, so it's reasonable to miss them. This work is accepted to the conference RECOMP, and we also published in the Journal of Computational Biology. The next project I'm going to talk about is the gender interaction detection using a 2-stage model. As we mentioned at the beginning, SNP is an important type of genetic variants. In this example, we have 2 SNPs. We can see that at this nucleotide, there are allele A and T, and at this position, we have G and C. And also, we are interested in some phenotype, for example, high cholesterol density level. And in this study, we focus on the continuous phenotype. And in the study, we are interested in which SNP is associated with the phenotype of interest. So we wanted to know which SNP is associated. The process to discover which SNP is associated with is called the genome-wide association study. And in the genome-wide association study, we will conduct a statistical test for each SNP. And on one SNP, we will split all the samples into 2 groups. All the individuals in one group will have the same allele on the investigating SNP. So in this example, we can see that all the individuals in the first group, they have allele A, and all the individuals in the second group, they have allele T. And then we compute the average phenotype value for each group, which is 1.2 for the first group and 0.2 for the second group in this example. And then the difference between these 2 average phenotype value will be used as a test statistic. So if the test statistic is greater than a predetermined threshold, we claim that the investigating SNP is associated with the phenotype of interest. However, recently many studies show that the phenotype value may be influenced by a pair of SNPs together. So the phenotype will only be changed when both the SNPs have the causal alleles. In this example, we can see we have SNP A and SNP B, and the causal allele for SNP A is A, and the causal allele for SNP B is G. So only when the individuals have this causal allele and both SNPs, their phenotype value will be increased from the other individuals. And following the same strategy with the GWAS study, we split the individuals into two groups. Now, all the individuals in the first group have coded allele and both SNPs, and the rest individual forms the second group. And then we compute the average phenotype value for each group and see whether the difference between the average phenotype value is greater than the threshold. This is called the pairwise association test. In this pairwise association test, we have to compute the test statistic, which is denoted as SAB, for all possible pairs of SNPs, which is the brute force approach. And we declare the SNP pair to be significant if the test statistic SAB is greater than the threshold T2. However, this method has great computational challenge because for the current GWAS study, it's common that we collect genotypes on 300,000 SNPs for 5,000 individuals. This is a normal size. And even in this normal size data set, we have to conduct 45 billion tests for these 5,000 individuals. This will take a really long time to finish. And the size of the data set is even growing. Here we define a concept called cost of our approach, and the cost is defined as the number of association tests we have to conduct. So for the brute force approach, the cost will be choose 2 from M, where M is the number of SNPs because we have to consider every possible pair. So could we find a method to do this pairwise association test much faster? Let's see if we have a significant pair AB, what will happen if we do the single SNP test on each of the SNP. Now let's see if we do the single test on SNP A, we split the samples into two groups based on the allele of SNP A, and we compute the average phenotype value, which is 0.7 and 0.2 respectively. Now the difference between the two average phenotype value is 0.5. We can see this difference is not 0, and it's not as big as 1, but still we can see there's a difference from the null hypothesis, which should be 0. And let's look at SNP B. Again, we split all the samples into two groups, and the average phenotype value for the two groups are 0.9 and 0.2 respectively. So the difference is 0.7. Again, this difference is not as high as 1, but also it's not 0. So based on this observation, we can see that if a SNP pair AB is significantly associated with the phenotype, then each of the SNP will have a marginal effect. So based on this observation, we proposed a threshold-based efficient pairwise association approach. We call it TAPA. The intuition of this model is that each SNP in a significant SNP pair has a weak marginal effect. And in our method, we have two stages. In the first stage, we propose two thresholds T and TB. For all those SNPs that pass the threshold TA, we put them into a set A. And for all SNPs that has the statistic greater than TB, we put them into a set B. Then in the second stage, we will pair all SNPs from the first set with all SNPs from the second set, and we calculate the test statistic SAB for each of such pair. Now we can, if the set A and set B is small, we can greatly reduce the number of tests, that is the cost. So our goal is to minimize the cost. However, we also want to guarantee that our method is not very different from the brute force approach. We want to guarantee that we discover 99% of the significant SNP pairs discovered by the brute force approach. So the crucial part will be how to choose the two thresholds T and TB to minimize the cost while guaranteeing the power. We will test a SNP pair only if the statistic SA is greater than TA and SB greater than TB. So imagine that if we set T and TB both to be 0, then every SNP will pass the threshold. That means we will test every possible pair. So this is same with the brute force approach. And here, we introduced a concept called power to assess the performance of an approach. The power is defined as the probability of discovering a SNP pair as significant if the SNP pair is really significant. So for our method TEPA, the power will be the probability of SA greater than TA, SB greater than TB, and SAB greater than T2, because that is the only condition that we were able to declare SNP pair AB as significant. In order to compute this probability, we have to know the joint distribution of SA, SB, and SAB. These three variables are not independent because, for example, for variable SA and SAB, they all involve the SNP A, so they are correlated. Fortunately, we can use a multivariate normal distribution to approximate this joint distribution. So now we are able to compute the power of TEPA. And also, we know that the power of the brute force approach will be the probability of SAB greater than T2. Compared to brute force approach, the power loss of our method TEPA will be 1 minus P power of our method over the power of the brute force approach. And now let's see the cost of our method. Remember that the cost is defined as the number of association tests. So for our method, the cost will be m squared times alpha A, alpha B, where alpha A is the significance level corresponds to TA, and alpha B will be the significance level corresponds to TB, because then the size of set A will be m times alpha A, and the size of set B will be m times alpha B, because most of the SNPs are not associated with the phenotype and they are under the null hypothesis. So the cost saving of our method compared to the brute force approach will be 1 over 2 alpha A, alpha B. Now we are able to compute the power loss of our method. We are able to compute the cost saving. So in order to achieve our goal that we guarantee the power loss to be 1% and minimize the cost saving, we can search over the space of T and TB to select the best T and TB that has the power loss of 1% and the cost is minimized. We applied the framework to a data set which is the North Finland birth cohort data, which has 5,000 individuals and 330,000 SNPs. We investigated the phenotype C-reactive protein, and we ran the method on 2.3 GHz AMD Opteron processor. The method TEPA, which is our method, takes 24.5 hours to finish, and the brute force approach takes 1,542 hours. So we can see our method is really 63 times faster. Now let's see whether we really achieve 1% of power loss. We designed a simulation framework to verify the power loss. We implant a real causal SNP pair into the NFBC data, which is the North Finland birth cohort, and we update the phenotype for each individual according to whether they have the causal alleles on both SNPs or not. Then we run the method TEPA and brute force approach to this data set. We repeat this experiment 10,000 times, and we see that the power loss of our method is only 0.4%. So our method has the desired power loss, and it's much faster than the brute force approach. This work is also accepted to the conference RECOMP, and it will also appear in the Journal of Computational Biology.
If you wanted to increase the accuracy, could you just sort of change the parameters for the method you're using? I mean, so you have 0.4%.
Yes, right. Yeah, we can.
Can't you just adjust the significance levels a little bit and then improve it?
Yes. So if we want to have like a 0.1 power loss, we can make the T and TB higher. Lower.
Right.
Yeah.
And it wouldn't be much more expensive, right? Or would it?
Yes, right.
A few more hours.
Yeah, a few more hours, right. We can adjust the T and TB to get a balance between the power loss and the cost. You mean for this, we use the z-test. We compute the z-value. Theoretically, we only prove this z-test has this power loss and cost saving. For other tests, it's possible that we may have similar conclusion, but we have to derive the equations to that specific test. Any questions?
When you say 10,000 times,
Mm-hmm.
you run, what does that mean exactly? You didn't run 1,500 hours?
No. Because we only needed to test whether we are able to find the real causal SNP. So we can only make the data set 2 SNPs. So now I'm going to talk about the last project, which is detecting IBD segments associated with quantitative traits. IBD means identity by descent. So 2 individuals will be IBD at a locus if their allele is inherited from the recent common ancestor. In this pedigree graph, let's look at these 2 individuals and the last generation. And we are interested in this brown region. We can see that the brown region are all inherited from the same person here. So these 2 individuals sharing IBD status and this brown region, and this brown region is an IBD segment. It will be very interesting if there's a disease-causing mutation in this IBD segment, because if this segment has the disease-causing mutation, then all the individuals that share IBD status will have similar disease status or phenotype value. Many methods have been proposed to detect the IBD segments associated with a certain disease or trait, but most of those methods are designed for case-control study only. And in this study, I developed a method to detect the IBD segments associated with quantitative traits. So next, I will show using an example how our method works. In this example, we have 8 individuals, A, B, C, D, E, F, G, H, and there are many methods that have been developed to detect the IBD segments from a data set. And here, at a certain locus, we have the IBD information as shown in this table. We can see that in this at this locus, individual A and B, they are IBD. Individual A and C are IBD, B and C, D and F, G and H, they are all IBD. And we construct an IBD graph from this IBD information. In this IBD graph, each vertex represents 1 individual, and we build an edge between 2 individuals if they share IBD. So you can see that there's an edge between AB, AC, and BC. For each node in this graph, there's a phenotype value associated with this node, which is the trait value. And as we mentioned before, if the IBD contains the mutation, contains the disease-causing mutation, all the individuals will have a similar phenotype. So for example, in this clique, all the individuals, their phenotype will be, let's assume the phenotype is higher if they have the causal allele. So the phenotype of all individuals in this clique will be higher than the other individuals. So we want to capture this property, and we design a test statistic to see whether the IBD segment is associated with the quantitative traits. And because in a clique, if there are more individuals sharing IBD, there will be more edges in this clique. So it's natural that we develop a statistic according to the edges. So for each edge, we get a summation of the phenotype value associated with the edge on the 2 nodes, and then we sum over all edges, and this will serve as our test statistic. However, because this test statistic depends on the edge, which is determined by 2 individuals, it will be really hard to derive the asymptotic distribution of this test statistic. So we have to use permutation test to assess the significance level of this statistic. So we will permute the vector of the phenotype, and for each permutation we compute the statistic based on this permutation, and this will serve as the null distribution of the test statistic. And the p-value will be the number of null statistic greater than the observed statistic from the original test divided by the total number of permutations. However, in order to obtain a small p-value, which is required in GWAS study, we have to make the denominator large, which is we have to permute a large number of tests. So the first two steps will cause a lot of computational time. Then we wanted to design a method to make this method much faster. Now let's look back to the edge-based statistic, which is summed over all edges here. And we split each edge in this graph to make another graph here. And in this graph, each node will be adjacent to a bunch of edges. And the number of edges adjacent to each node will be the number of individuals that are IBD with the individual. So we can define the number of edges associated with each node to be the IBD degree type. And then we can see that in this test statistic, each phenotype value will contribute to the total statistic DI times, where DI is the IBD degree type in this graph. So we can rewrite the edge-based statistic into this form, which is a weighted sum over the phenotype. Here, the weight for each phenotype is the IBD degree type. We can see that these two test statistics are exactly equivalent. Now we write it in the vector form, which is d transpose y. So we can see that this d transpose y is highly similar to the estimate in a linear regression. So under the assumption that the phenotype value follows a normal distribution with the variance sigma square, I develop a linear model here. So the yi is the phenotype value, and the di here is the IBD degree type. And this formula, we can see that the di is similar to the genotype in a normal linear model. And the error term follows a normal distribution with variance to be sigma square. And we write it in the vector form. We will be in this way. And using the ordinary least sums, we can estimate the coefficient of the di, which is gamma. So we estimate the gamma to be this formula. And under the null hypothesis, in which case the current locus is not associated with the phenotype, the gamma will follow a normal distribution. So we are able to compute the p-value of gamma, which is the significance level, and the null distribution easily. And now let's look back to our IBD degree type sum statistic. We see that these two equations, they are very similar. Here we have a term d transpose y, and here we also have a term d transpose y. And the only difference between the estimation of gamma and the test statistic Sk is a constant factor. So we know that under the null hypothesis, the Sk will also follow a normal distribution, and we are able to compute the p-value of Sk from the normal null distribution. Actually, because they only differ by a constant factor, the p-value of gamma here will be exactly the p-value of Sk. So instead of doing the permutation test to assess the significance level of Sk, we build the previous linear model and compute the p-value for the coefficient gamma. The p-value will be the p-value of this test statistical Eskin. We apply our method to the North Finland birth cohort data, which is the same with before. And here we show the results. In this figure, on the x-axis, we show the log p-value from 10,000 permutations. And in the y-axis, we show the log p-value from the linear model. We can see that the p-values are highly correlated. And here the red line shows the lower bound for 10,000 permutations because at the lowest value, the p-value will be 10 to the power of -4. We can see that no matter when the p-value is high or low, our method, the linear model, will always have a very similar p-value with the permutation test. And this work is just finished, and we finished the manuscript, and we are preparing to submit it. And now please allow me to thank my committee, Professor Eleazar Eskin, Professor Ernst, Professor Nielsen Freimer, and Professor Stott Parker. And also, I would like to thank my collaborators from UCLA and outside of UCLA. Especially, I would like to thank Professor Anand David, who is not here today, and Professor Christopher Lee. They give me a lot of comments from my qualified exam. And also, I want to thank my collaborators in the Eleazar Eskin's lab, Farhad Hamoudari, Joanne, Sergey, Michael Bello, and Yuan, who is now a postdoc, and Nick, who is now in 23 and me, and Emerald, who is at Illumina, and Dan He in IBM, Boom Jihong. And also, I would like to thank Professor Aaron Halperin and Professor Sergey Sner and Professor Jose Lozano. They collaborated with a lot of people with me, and they helped me a lot in developing the models. Also, I would like to thank Professor Lakshmi Preda in IBM. I did an internship last year with her in IBM research, and she gave me a lot of guidance. Also, I would like to thank my collaborators in Jake Lucy's lab, including Matty and Brian. And also, I would like to thank the architects group in UCLA, with whom we have some collaboration going on. Professor Jason Kong, Professor Glenn Raymond, and my collaborators Yuting Chen, Peng Wei, and Marco Vitazar. Okay, thank you very much. Any questions? Mm-hmm. You mean a segment of DNA which has the same haplotype between individuals? Well, here we allow that the IBD segments, there might be small difference between individuals. And the reason that we wanted to do the IBD association mapping is that it may harbor the rare alleles. But for haplotype mapping, it cannot capture the rare variants. Because if the it's especially useful for rare variants because the rare variants may be introduced into the population recently. So there's only a small portion for the individual has the rare variants, and these rare variants might be captured by the IBD segments. And the more recent, the longer the IBD segments, and it's much easier to capture it.
So how do you generate the IBD and get the IBD?
There are a lot of programs that can detect IBD segments, like Beagle, Germline. They can detect the IBD with a high accuracy, even as short as 2 centimorgan.
So then another part of my question is that I understand that the concern about being able to capture the effects of rare variants.
Mm-hmm.
Other than that, let's say we were able to capture that within our information, right?
Mm.
And IBD or haplotype are all required, right? So it's being captured. But if you look at this haplotype map,
Mm-hmm.
So, could you tell me what's the rationale behind it? You want to come up with something?
Like I explained here, the individuals here, they might have a similar phenotype, right? So the reason that we sum over edges to aggregate the difference of this phenotype that is different from the others.
To sum up the
Yeah, with
from your formula, it looks like for each edge you just added the
yes, we added
type of the two nodes, right?
yes, right.
And then you sum up the sum of the node being added twice.
Yes, it may even more times.
Well, more times because
Yeah, right. That's the
you
int.
have like a click fully connected like
Mm.
bigger than 3, right?
Yeah,
So you'll be added many more times.
yeah, many more times. That's why it will be different from the others, because one difference, the difference, for example, here from the others, for example, is higher than the others by 1. And if we add it 10 times, the difference will be enlarged by 10 times. So the more connected the clique, the higher our test statistic is.
So you added up the test statistics or you add up the phenotype?
We end up the phenotype.
So what if like say for this click,
Mm-hmm.
my phenotype value are small, right?
Mm-hmm.
And for another smaller click, my phenotype value is large, right? So when you add them up,
Mm-hmm.
because you add them more times in this big click,
Mhm.
right? And then the small value all add up maybe would equal, or roughly equal the small click because each node have a larger phenotype value. Then in the end, perhaps it's
Yes, right, exactly. That's why we see that our method is able to capture the rare variance which happens recently. So if the rare variance happened recently, so there might be only one click. There's not the other click. Because only this click, they will share IBD status.
well, but the rest of them,
Yes, the rest they may share, but their click may be small, and they don't have the causal mutation, so their phenotype won't contribute too much to the test statistic. Because if the click is large, then the phenotype will contribute more times.
so I guess maybe this is because often I don't
Okay.
quite understand why the other part is not able to form large click.
Okay.
It could also be empty, right?
Yeah, they could, but the reason is that the others will be small, the other click.
Okay, anyway.
Okay, we can talk after. Yeah.
And then second part,
Mm-hmm.
can you clarify sort of what's the approach for handling the multiple testing issues with
Oh.
errors in terms of the statistical?
You mean the multiple testing, right?
Yeah.
So to handle that, we use a very low, very high T2 that is corresponded to a very stringent significance level, like 10 to the power of 9, minus 9, or 10 to the power of minus 10.
So, I mean, does this correspond to some sort of Boelter inspection?
Actually, it's not as strict as Boelter correction, but from the empirical research, many people use the p-value to be threshold to be 10 to the power of minus 9 or minus 10.
Is there any theoretical reason for this?
I think at the beginning they use like 10 to the power of minus 12, which is the Boelter correction, but they fail to discover any significant pairs. So they relax this constraint and find some significant pairs.
Well, I don't understand that. I mean, by relaxing the constraint, are they really finding significant pairs, or are they just relaxing the constraint?
Yes, I remember there's one paper they used 10 to the power of minus 9, and they really find some real causal pairs. It's published in...
How do they define that then? Some sort of circular?
I'm not sure why they pick up this value, but I think according to my understanding, they rank all the pairs and find the most significant one, and then they validate that one.
There's a lot of LV, not LV, but correlation between statistics when you're looking at multiple
actually can compute it. No one can actually do permutations, right? And compute all the pairs, because you can just compute it as soon as it's one takes like months.
Yeah, but, so did you compare with our
You mean team?
Nova? No, faster Nova.
Fast Nova?
Yeah, one condition you can have.
Yes, but I didn't remember whether ANOVA can work on the quantitative traits. I remember that I tried to run the fast ANOVA, but there was some problem. I emailed Zhanyong at that time, and we didn't figure it out. So finally, we didn't compare with it. So I worked with Zhanyong, email back and forth, want to make the fast ANOVA work on our data set, but we didn't make it work.
Okay, I'll help you.
Okay, okay.
Thank you so much.
So I had a question about the IDD
Mm-hmm.
approach. So you mentioned that, where there's truly an IDD with a rare variant, let's say, that's contributing to the phenotype,
Mm-hmm.
you expect there to be a longer IDD segment.
Yes, for if we aim to capture the rare variants.
Right, but I may have missed, where does the length of the segment actually come into your approach? I didn't actually see that you're actually assessing the length of the segment.
You mean the length of the IBD segments?
Right.
Well, the length is, I don't have the exact number now, but I remember that the length is not very long because for the current IBD detection method, they did not do a good job in phasing. So, for example, in this figure, sorry. So, for example, in this figure, these IBD segments, they predict to be from the same chromosome here, same strand. But it's also possible that they predict half of them to be from here and half of them from this strand. And they didn't do a good job in the phasing. So it's possible that we have long segments, but it displays it in short segments.
Okay, so you don't actually have a measurement of the length of the segment?
I remember the average length is like 10 or 20 centimorgan. No, it's not uniform.
Yeah, I think as you were saying, the expectation is that where the IBD segment is because of them sharing the causal allele as opposed to the relationship being close, you should have a longer IBD.
Yes, right.
But again, it seems to me that it's important if you can have a method like this that you actually have a way of sort of quantifying that length difference.
Yeah, sure. Okay.
One thing I was curious about is that you mentioned the other paper, right? That is compare with the
Yes.
And what's the method?
They use, I forgot the name of their paper, but their method use a similar strategy, which is based on the read depth, but they didn't consider the multiple mapping rate. That's why they can only see whether it's copy gain or copy loss. And they miss some region compared to our method.
So the difference here so the advantage of your approach is that you can represent how many copies are there?
Yeah, we can exactly see how many copies are there, and we can predict the boundary very accurately.
So they also use the EM method?
No, they didn't use EM method. Roughly, the idea is similar to the traditional approach, but they will split the reference genome into bins. For example, 1,000 kilobases, and they compute how many reads are mapped to each bin. And then, if for a bin the read depth is higher than the other, they will see this is a copy number variation, and they will connect the adjacent bins if they all have this CMA stat. I think they have it. Yeah, they have the Finnemark model. Yeah, many of them, yeah.
Many of them based on.
Yeah. Actually, we also, yeah, right, because the assumption is that the adjacent beings, they will have a similar copy number. Actually, in our method, we also use a hidden marked model, but I didn't explain here because it's too complicated. Not beings, we based on nucleotides. Yes, that's why we have high resolution.
I see. Do you consider like easy content?
Yes, right. So after we here, when we assign the reads, we will correct the GC content. So we use a formula to adjust the GC reach region. The read depth will be adjusted according to the global depth and the region with the high GC content.
Can you compare with Eskin, another software that came out last year to
This paper is published in 2012, so we compare with the CNVeM, which is the state-of-art method at that time.
So is that result in
No, that result is not from CNVeM.
so how
I didn't show it here, but compared to CNVeM, our method is more accurate. So compared to CNVeM, suppose this is the result of CNVeM, and our method will roughly have this, like this size, is a subset of CNVeM, because CNVeM has lots of false positives. You mean the false positives? Because we did on the simulation data set, so we know which one is real. Yeah, this is on real data. We test our work on both the simulation data and the real data. We compare our method with the CNVeM on simulation data, and we see that our boundary is much more accurate than CNVeM, and we can see the copy number while CNVeM cannot.
Question. I mean, you've got many algorithms in the thesis for copy number variation and analysis, and they seem very practical. Lots of iterations and can be used.
Mm-hmm.
But then you stop with like one data set or two data sets or something like that. And have you thought about more, trying it out on more data sets just to see what happens and maybe other results?
Yes, right. For example, we test on the mouse data set, we test on the human data set, which is not banana birth cohort. We also test on the HMDP data recently. And
okay, I'm not criticizing, I'm just wondering, since it seems practical and you did much better than CNVeM for this particular thing.
Yeah.
You could try it on other things, right?
Yes, sure.
It's not that there's not enough, it's just that it looked like you could get a lot more mileage out of it.
Yeah, right.
But so then you're not going to.
Yeah, we didn't test on more data set yet.
Is there a reason, or is it just that you didn't have time, or why?
Well, I think the reason is that first so our method will work well on the mouse data. We didn't test the CNV method on the human data, so it's hard to see because for mouse data it's the inbred mouse, so it's haploid, and for human it's diploid. There are two strands for each chromosome, so the performance might be different. So theoretically. Yes, right. Because CNVeM, they don't predict the exact copy numbers, so they only have to know whether it's copy gain or copy loss. And for us, yeah. Yeah, actually, I tested the method on one of the it's a cancer data, and I think compared to CNVeM, we also have some. We also have less false positives. And I didn't put it in the paper because that's after the paper published. I tried it on cancer data, and but we didn't publish that one.
We actually reached out and we tried to collaborate with the incubator so that they would replace their algorithm. But it just kind of didn't work out. I mean, the reason practically, there's so many filters No, I mean, seriously, you know, like,
Yeah, no,
or
no.
we want to work with CNV data to get our stuff into their code base. And so, you know, we reached out, but, you know, all the methods are still pretty noisy, and none of them work very well. It wasn't like this, having a better algorithm was really a problem, as long as the data is not very good.
Yes.
This
Yes.
And also, the data that you guys have now is much cleaner, right? Like, coming off years of latest genomes. Yeah, so, you know, that's part of it, that's part of the issue. Any more questions?
Okay, so let's thank
Thank you.